In this case, in the left column, there's a dipole bilateral thalamus. The right column is bilateral thalamus, but also right occipital. So it's an actual source of that. Then we compare 19 channel EEG to 128 channel EEG, and you get essentially the same resolution. Okay? 128 channel EEG costs over $100,000, and 19 channel EEG, $3,000. Magnetism, it's a 148 channel EEG device, gives you a little bit better resolution, but again, it's not portable. You have to have liquid medium and even shield the room. So EEG itself will resolve the sources for 19 channels just about as well as 128 channels. When I worked at the National Institute of Health as a project manager, we compared 19 channels to 32 channels, 64 channels to 128 to 256. Essentially, you do get an improvement from 19 to 32, slight improvement, but beyond that, there is not much improvement. And quite a bit of money went into that process, and we're glad to see that, because part of the secret is that the brain's organized in these clusters, in these groups. And what we do now is we measure the electrical sources from single voxels inside we got 12,400 voxel MRI, but we know where the Brodmann areas are, we know the center of the Brodmann areas, so we measure the current coming out of the center voxel of each of the Brodmann. And then we measure the time it takes to go from one center voxel to the other. It's called coherence or phase delays related to the conduction velocity of the white matter and the speed at which neurons can synchronize. Now, one question that arose during all of this was, is it possible to measure the cerebellum sources, which are of interest to this group here especially? And so if you do a search in the National Library of Medicine and type in the search term EEG of cerebellum, you'll find 2,036 citations. There's more now. This is a year ago that I put the slide together. And so EEG is capable of measuring the sources as deep in the brain as the cerebellum. And this is just a quick anatomical view of the cerebellum, really to know all of that. But this is a little bit more interesting in that the green diagram on the left shows the mapping of the body to the cerebellum. The flocculus nodulus, the blue on the bottom, is where the vestibular nuclei project to determine your balance. And now the body surface is cerebellum is made up of these different parts. But if you look at the topology of the body surface at the top there, you'll see the head is upside down, the hands are spread out, the feet are large. The body doesn't have very big representation because there's not as many nerves in it, but your hands, your thumb, your face are very well mapped in the cerebellum. Now, if you look at the bottom, you'll see the body has been split in half and rotated 90 degrees. So it's a 180-degree flip, 90-degree rotation. And we discovered in the late 1800s, really, and it's used throughout physics, that if you take an object and rotate it 90 degrees and flip it 180 degrees, it improves the signal-noise ratio of the DNA molecule, which is an example of the fiber for the replication. And we know too that in the retina, for example, of the disc, it maps to the visual cortex, which is essentially a rectangle. And the mapping of the disc to a rectangle is called a conformal map. And the advantage of the conformal map is that you've got size invariant, rotational invariance, you have computational neuroanatomy by virtue of that map. Now the brain doesn't operate like a computer does. Okay? It does its computations by mapping and remapping. The culprit is the logarithmic spiral. Oh, by the way, the conformal map has two extremes. If one extreme is zero, it's like if you drop a pebble in the bathtub, you get concentric circles going out from the point of impact on the water. And then the other extreme is infinity, which is like a firecracker going off, an explosion. Radial lines shooting out every direction. And everything between 0 and infinity is a spiral. It's a logarithmic spiral. It's by having a logarithm, one of the computational advantages, that you get multiplication by addition. That's what logarithm does. In the 1500s, when we started developing logarithms, it was a big important advancement in mathematics for that reason. So the brain is doing computational neuroanatomy. The retina maps to the visual cortex, and the mapping itself does the computation. Computers are not that way. It doesn't make any difference if you got a big case in your computer or a small case, or the shape of the computer is irrelevant. It works with registers, zeros and ones, and CPU and all that kind of stuff. So the brain is just mapping and remapping all the time and doing computation. So color, shape, form, movement, all of that is computationally computed by the neuroanatomy itself. And you can see that in the cerebellum. This replication, this flipping, are an example of the conformal map. So we looked into the sources of the cerebellar sources at the scalp surface to see what extent can be measured then. And we have a very large FDA registered normative database from birth to 82 years of age. And we wanted to explore this and be able to compare individuals to this reference database. In quantitative EEG, reference databases, if they're good quality reference databases, are similar to databases in medicine that's used all the time, blood tests, for example. You don't know exactly whether your cholesterol is too high or low, or your adequates are too high or too low, or etc. Only way you do is because there's been a reference database of healthy people, and then you know your adequates are too low or too high. And you need reference databases, numerous reference databases in medicine, and EEG is one more. But very important to have. Now this is an example of a study showing sources from the cerebellum when we're measuring EEG at the scalp surface. And you can validate and verify those as part of those 2,000 studies I mentioned. Now this is another study. This is what standardized weighted Loretta, where people imagine moving their left hand or their right arm or their left foot or their right foot, etc. And just imagining it allows you to see sources in the arising from the cerebellum. So our brains are actually predicting where things are, where they're going to be in the future. The cerebellum has about 80 billion neurons. Cortex, roughly 100 billion, but the cerebellum is 1/20th the size of the cortex. Very, very special organ. Very, very important. And it has no direct connections. All of its outputs and inputs go through relay nuclei. And one of them is the red nucleus I'm going to show in a minute that can be studied, that actually goes from the cerebellum up to the frontal lobes as well as down to the spinal cord. So the cerebellum is bridging the gap between the lower body and your frontal lobe. This is a study by Samuelson et al. Where they wanted to look at the sources of the electrical sources coming from the cerebellum, measuring it at the scalp surface. And they discovered that they could easily measure it, that it's a weaker signal because it's some distance away from the scalp surface. And so it's got a stronger signal the closer you are with that pattern that comes up to near that surface, as I mentioned from the 1800s, versus the deeper sources. But nonetheless, because the sophistication and quality of the amplifiers we use today, we can easily detect these relatively small signals. And this is from the Samuelson paper, where they looked at the cerebellar sources and the cortical sources using magnetic encephalography as well as quantitative EEG. And they found that the cerebellum, which is in blue, they can detect the sources, but it decreases as a function of distance. And they compared different types of magnetic encephalography to quantitative EEG, and to look at both cortical sources and cerebellar sources. And this is the results of the magnetic encephalography in which the colored code tells you the strength of the signal coming from the cerebellum using magnetic encephalography. And you can see different parts of the cerebellum, the vermis and the center parts and the different lobules of different strengths. And they can pick up the signal almost everywhere from the cerebellum. The blue, it shows a weaker signal compared to the red, but they can still detect it. So this is the EEG source, which is actually a little bit stronger than the energy source. And so using an EEG, you can look at these different parts of the cerebellum, perhaps you can see from the surface of the cortex, and can be verified as coming from the cerebellum.